just start the recording and then instruct you guys. Okay, so today we have group 1 and group 2 to present. Please note that yes, I think Nitin sir has asked a question in the chat box. You can answer that because you have studied that in Inspire. So put your answers there in the chat box. So we will have presentations for group 1 and group 2. The presentations will be recorded. Okay, one of you can cast the screen and the others can, come turn by turn and speak. That's completely fine. So it's up to you who wants to cast the screen and go ahead. Okay, that decision is going to be yours. There's no need to be worried. Keep calm. Say whatever you know. Okay, it's a learning process. And also, other students here are free to ask questions. It's essential that each one of you is going to ask questions over here. I don't want you to protect each other. That's not going to be anyone's benefit. It's going to be a loss instead. Okay, so please see to it that you are asking questions and let it be a healthy discussion wherein you learn things in the process. Okay, so over to the first group, we have Akhil, Akshay, Harshali, Sarvesh, and Preksha in group 1. So over to you guys.
Yes.
Okay, so as you present this, whoever is presenting it could zoom at the part where this person is going to speak. Okay, so please.
Yes, sir. Yes.
Can we start?
Sure.
Okay. So good morning to one and all present here. I am Harshali Avai, roll number 7503, and today our group, which is from roll number 7501 to 7505, are going to present a poster on the upper respiratory tract, the infections associated with it, the samples which can be collected, how they can be processed, the disease can be diagnosed, and also some preventions and precautions to avoid this infection. So now, without further ado, let's dwell into understanding our upper respiratory tract. So the upper respiratory tract or the upper respiratory system consists of the nose and nasal cavity, the larynx, and the pharynx. These organs allow us to speak and breathe. These structures, uh, these structures filter out, clean the air we inhale, including the smoke and other pollutants before the air travel down to the lungs. Now, let's talk about the infections associated with the upper respiratory tract. So, without a doubt, the common cold is the most well known URTI, that is, upper respiratory tract infection caused by the rhinovirus. Some other common URTIs include sinusitis, laryngitis, epiglottis, and bronchitis. First, let's talk about sinusitis. Sinusitis is the inflammation of nasal sinuses. Sinuses are basically the hollow spaces within the bones between your eyes, behind your cheekbones, and in your eyes. They make and in your forehead. They make mucus, which helps the inner organ to stay moist. Often, sinusitis is caused by the bacteria Streptococcus pneumoniae, known as the strep throat. Next is the epiglottitis. Epiglottitis is the inflammation of epiglottis, the upper part of your trachea. It protects the airway from the foreign particles that could get into the lungs. The swelling of epiglottis can be dangerous as it can block the flow of the air traveling down the lungs. Just a minute, sorry. epiglottitis is often caused by Haemophilus influenza B bacteria. Next is the laryngitis. Laryngitis is the inflammation of larynx or the voice box. It is usually caused by the straining or overusing your voice cords by speaking or yelling. Just a minute. Sorry, I'm making a call. So, laryngitis can also be caused by the bacterial infection sometimes and also by drinking too much alcohol. Next is the bronchitis. Bronchitis is the inflammation of the lining of your bronchial tubes, which carry the air to and from the lungs. People have bronchitis often cough up thickened mucus. Chronic bronchitis is usually, but not always, caused by smoking tobacco. It can also be caused by exposure to secondhand cigarette smoke, pollution, pollutants, and toxic gases. Bacteria can also cause bronchitis with people underlying health problems. Mycoplasma pneumoniae, Streptococcus pneumoniae, Haemophilus influenza are most commonly involved. Now, there are some common symptoms which can be which we can from which we can identify this URTI, like runny nose, nasal congestion, mucus production, and other symptoms also include fever, fatigue, pain while swallowing, and headache, etc. Now, my friend Sarvesh will explain you the samples which can be taken and also how they can processed. Over to you, Sarvesh.
Okay, thank you, Harshali. Now, to move forward, I'll be talking about sample collection techniques. And to start with, the first and the most important point is the timing. The timing is a very decisive factor for the screening or to test the symptoms of any disease. So, it is said that if any person sees any symptoms of the disease, as Arshali mentioned earlier, the patient should visit the clinical facility within 3 days and not later than 7 days. So because the quality of the specimen is very decisive and accurate results. Moving forward to the requirements which we require for the testing. First is the swabs. So swab types. It is preferred that plastic or metal shaft swabs should be used. One second, sir. I'm getting a call. Sorry. Swab. So talking about swab types, we should use plastic or metal shafts, and normally we should avoid using wooden shafts. And the swabbing material which is there, it should be a flock foam and polyester such as Dacron or rayon. It is said that cotton and calcium alginate should be strictly prohibited because they may interfere in the molecular assays such as PCR and PCR. First of all, to explain the specimen collection techniques, there are three types. First is the oropharyngeal swabbing technique. Uh, Oropharyngeal swabbing technique is very simple process in which the patient is said to have his posture accurate appropriately. He is said to open his mouth, and first of all, the one who is assisting him, the person who is taking the samples, he should first of all sterilize his hands and use all the proper equipments, such as gloves and all mask, etc. And the patient is said to open his mouth and create an ah sound so that he completely opens his mouth. proper lighting conditions should be provided so that whole of the buccal cavity can be seen clearly. With one hand, a tongue depressor is used, and with the other hand, a swab is inserted into the nasopharyngeal region. It is to be seen that the swab doesn't touch the cheek cells or the tongue, so that the normal flora doesn't come into contact with the swab. This type of technique is normally used for collection of adenoviruses, herpes viruses, mycoplasmas, chlamydia, candida species, and corynebacterium diphtheria. Moving to the nasopharyngeal swabbing, it is kind of similar to oropharyngeal swabbing, in which the only difference is that the swab should be flexible. It is not a rigid swab. Okay, so the swab should be flexible and it should be inserted into the nasopharyngeal region through the nostrils. And common goal here is to collect the ciliated columnar epithelial cells, which is to collect them. And talking about the procedure, the swab should be around kept in the that area for 10 seconds and should be moved very slowly so that all the secretions are collected appropriately. This technique is also suited for Mortadella pertussis, Neisseria species, respiratory syncytial viruses, parainfluenza virus, and virus causing rhinitis. Moving forward to the last step is collecting the nasopharyngeal washes or the aspirates. This technique is commonly used for children or infants, but not necessarily. We can collect it from adults as well. This technique is commonly divided into two parts. There are two methods for this technique. First is mechanically, and other one is non-mechanically. Non-mechanically is comparatively easier. I'll first try to explain the mechanical suctioning of collecting the aspirates. As you can see in the image on the left-hand side, on the right-hand side, it is shown that there is a vial through which there are two pipes connected. One pipe which is shown one pipe is going into the nostril of the person, and the other pipe which goes into a pump. The pump is set at around 80 mm of Hg pressure, so that it creates a optimal suctioning for collecting the samples. Before that, the person puts 2.2 to 2.5 ml of sterile saline into the each of the nostrils of the person who is the patient, and then the pipe is inserted into his nostrils, and both the specimens can be collected into that vial. Both the specimens from both the nostrils can be collected into the same vial. And as we spoke about collection of the samples, that is the swab types or the liquid samples, they can be collected and sent to the laboratory for further sampling. And it is said that if the samples are going to be tested immediately, it can be stored in a it can be stored. It is okay if it is not stored, but if it is going to take more than 24 to 48 hours, it is necessary that those samples should be kept in a appropriate transport mediums, which Akhil is going to now explain in the next part. So thank you.
Thank you, Sarvesh. I am roll number 7501, and I'd first start with a general note on sample processing methods. It is often said that the swabs used for sample collection should be immediately placed into a sterile tube or other respective containers, which include the required media or external factors depending on the sample culture. It is also said that it should be desiccated with materials such as silica gel so as to minimize the presence of many of the species that are in the sample. Uh, but exception for the desiccation would be group A streptococci, for example, Streptococcus pyogenes, which can live on the dry swabs for a time ranging from 48 hours to up to 72 hours. Uh, if the sample is collected and is going to be cultured under the 4-hour time frame, no extra precautions or many conditions would be necessary uh, to keep it viable, to keep the sample viable. But if the transport would take around 4 to 24 hours, it is often preferred that we use Amy's medium with charcoal or Stewart's medium. These media are used to maintain the viability and to control the overgrowth of the sample. If the transport would take longer than 24 hours, then Regan Loewe or Jones Kendrick medium can be used. These media are especially useful since they contain charcoal and starch, and it also contains cephalexin, which inhibits Haemophilus influenzae, some coliforms, and most streptococci, with the exception of enterococci. If the delay is going to be if the delay is going to be much lengthier, then Regan Loewe maintained at 4 degrees Celsius is preferred. Um but if you want to have the recovery of viral agents, special transport media is required, which is called as a viral transport media. But there are microbiologists who believe that viruses can be grown on bacterial transport media too. So for better ease and generalization, they created a universal transport media. This UTM is FDA cleared collection and transport system suitable for collection, transport, maintenance, and long-term free storage of clinical specimens like viruses, chlamydia, mycoplasma, and ureaplasma. These UTMs are often these UTMs come in plastic screw cap tubes and can and can maintain the viability for up to 48 hours at room temperature or refrigerated temperature. So further transportation details and diagnosis would be handled by Akshay. So Akshay, if you would. Thank you.
Yeah. Thank you, Akhil. So basically now, since they have collected the sample, now what is our main aim? Diagnostics, right? So we have to transport this sample very safely as possible. So we will have to take a very leak-proof container, sterile container in which we will have the sample. And after collecting, it must be transported to the nearby microbiological lab immediately at RT, or we can store it as per our need. But we should always remember first that the delay in the transportation will also lead to delay in results. So we have to plan that. So time is really crucial. So here it is mentioned, if you can see, the transportation should be done within 2 to 4 hours if maintained at RT, and 24, we can keep it for 24 to 48 hours if we are keeping it at 4 degrees Celsius, or if we are keeping it at minus 70 degree, we can stay for much longer. So for shipment, in case of shipping of the samples, if we have to store it for a very long time, so we basically use the gel cold, like hardened ice cold gels, so in which we can transport the samples safely. Now, if I talk about some hazardous viruses, so the shipping has to be done very carefully. So if you can see here in the image, there is something known as the triple packaging system. So I will just zoom it for you. So you can see there is a primary culture container. So there, as the name suggests, that it is a triple packaging system. So inside the primary culture container, you will place your sample, and you will place this entire container inside the secondary one. So we should make sure that the sample doesn't leak, break, or they puncture out into the secondary container. And the outer cover is for the shipping treatment. You will have the hazardous sign and other details. Now, this all were for like transportation. Now, for local transportation, if you can see the another diagram here, there is a bag. Inside it, there is a lot of materials. So basically, locally, you can just take the sample You can transfer it to transport media or buffer as mentioned by Akhil, and we can keep inside and we can transport this back safely to the nearby lab through vehicle or manually. So now after that, for ice cold storages, I would talk about. So there has to be a lot of layers of packing. The primary container will have the sample. You will keep inside a secondary one. Now this secondary, the whole apparatus is kept inside a Ziploc pouch, and it is completely sealed from top. And this whole substance is kept inside a thermocol box. And inside that we have the hard frozen gels. So for processing, maybe some diagnostics may require the genetic material of the organism. So for example, if you want to take some RNA, for example, so we will have to also lyse the cells. So after collection and storage and whatever the procedures, what we will do, we will use some lysine media or lysine chemicals, which we will get from this kit, as you can see here, or we will buy. So we can use the chemicals, we can lyse the cells, we can centrifuge, we can extract the genetic material, and we can use it for the immediate experiments, or you can store it. So if we are storing it before experimentation, we have to keep it for RT, we have to vortex it so that the sample becomes homogenized, and then you can use further. So in case of hospitals talking about, you get a variety of samples. So we have to have a very deep knowledge about what samples, when to process, what to process. So all the details with exact labeling, it is very necessary. So after transportation, we have the last step, that is the diagnostics. So diagnostics is very crucial because after diagnosing the infection and the causative agent, we will be able to tell that what treatments can be done with the patient. So the basic first thing which come to our mind is definitely the plating, the agar. We will isolate the organisms. So if you see in the left-hand corner, there is a red plate blood agar, which is used for the streptococcal species. We will have a chocolate agar, which is used for Neisseria and Haemophilus that are very fastidious. It is a very nutritious medium. And if you see in the middle, that is a telluride blood agar. It can be used for detecting many species. So it is a very fastidious type of media. So Corynebacterium, diphtheriae are the examples of organisms which can be isolated from this. So, but it is very necessary to understand that these plating processes take a lot of time, and we can't just depend upon only the plating. So there are various rapid and fast techniques which can be done along with the plating. So for example, if I talk about the rapid strep test, we detect the antigens which is present on the bacterial surface, and so we will put specific antibodies, and if the antigen antibody complex is formed, we will be able to see the color due to the chlorochromogenic substance used. Now there are various molecular techniques also. For example, real-time RT-PCR, you all must be very familiar of. So we will take the genetic material, we will use various primers. I will not go deep into the exact procedure, but there will use primers, quenchers, and fluorescence, and we can also quantify basis on the intensity of the fluorescence. So there are also various techniques such as modern techniques, which is very sophisticated but easier to use, which is known as MALDI-TOF MS. So it can be used. It is the various ribosomal proteins inside the bacteria, if you know, is very conserved. So these special bacterial proteins or the ribosomal proteins, if you have to detect that, MALDI-TOF can definitely use some biomarkers and can differentiate at the subspecies level also. So in this diagram, in the lower diagram, you can see the bacterial culture is passed through MALDI-TOF MS, and due to this, we can even identify the organism, and we can also identify the AST, that is antibiotic susceptible testing. So instead of that, MALDI-TOF can also detect some biotype on the microorganism, and we can also know that if it is a drug resistant strain or not. So like I can even explain the brief working of MALDI-TOF, how it is done in the hospitals or labs maybe, but after this presentation if you guys want. So yeah, so we can also use some rapid test or biochemical test simultaneously side by side. So you will get a very small sample from the transportation. You have to strategically divide it into small vials and perform simultaneously all the tests until we arrive into the conclusion. So now, since you guys know about how to collect the sample and what are the various steps until the diagnostics, now I would request Preksha to just conclude by mentioning some of the precautions which has to be taken while collection of the sample and various other steps, and also mention some preventions just as a general awareness, so that we can be a little safe, what we can do to prevent these respiratory upper tract infections. So over to you, Preksha.
Thank you, Akshay. So now first I will speak about some precautions which we have to take for to prevent this upper respiratory tract infection. So the healthcare provider must follow some precautions. Like so, the healthcare provider must wear a mask all the time when they are in a healthcare faculty. So they also use personal protective equipment which includes N95 or protective gloves or eye protection, PPE kit, etc. So, and they must use a sterile swab for the collection of the sample, and it is important because it ensures both patient safety also, and it ensures the integrity of the specimen also. And we should kept all the used swab away from the sterile swab to avoid the accidental contamination also. And after the picking out the sterile swab from the container, we must close that container to avoid the contamination. And if the patients are self collecting a sample, for example, then we must give that patient swab only while using a clean and protective set of the gloves. And if the patients need an assistance, then you can guide that patient like how to do that, and then transport that swab and place it into a transport media or the transport device, and then seal it. And all the contaminated container equipment we should must kept in a plastic bag, and then seal that bag, and label that bag with the content of the material and the nature of that content, and also indicate that it is for the disposal purpose. So now I will speak about some of the preventions. So there are several measures to prevent this upper respiratory tract infection. So smoking cessation, regular exercising, balanced diet, stress management, all these things are important, and it strengthens our immune system also, and it reduces the risk of infection also. So there are some other preventions also, like for example, we should wash our hands for about 20 seconds or more with a soap and or with a gentle warm water. And we should reduce our contact with the person who is infectious. And also we should clean all the objects which is touched by that infectious persons, for example mobile, door handle, telephone, etc. And we should give them vaccine also for the certain flus, etc. And to prevent the spread of infection in a evacuation center, the staff should identify the person who are ill, and use the appropriate measures as soon as possible. And if that staff is not well, then give them excuse from the work for some days or week. Or and there are some other regular preventive measures also, like we should not use a cloth, or we should not share our cloth with anyone else, or we should not share a towel or the toothbrush, eating utensil, drinking container with anyone else. So these are all about the prevention. And at the end there are references. Yes, so these are our references. Thank you.
Okay, good job. Keep it up. But I would want to suggest few things to everyone. Also, you guys, before you submit your PDFs on Classroom, uh, include the institute name and the department name at the top, just below your names. You can write the institute name. You can write it as Department of Microbiology, SP Mandis, Ramnaran Roy Autonomous College, Mumbai. Okay. You just include that before you share the PDF or you upload the PDF on Google Classroom. Okay, please remember that, everyone in the group. So questions? It's up to you. If you want to insert, you are free to do that. If you want to put the logo, you can do it. That's not a problem. Questions?
Sir, can
No,
I
go ahead and
ask?
ask. You don't have to tell me.
Akshay,
Okay, so
please zoom in on the reference point.
Sir, I had question for Akhil that he said that John's Kendrick transportation media is ideal for transformation and it contains cephalaxin, which is an antibiotic medication against to kill bacteria, and charcoal.
Okay, so Anjana, to respond to your question, for the Jones Kendrick medium, charcoal and starch are used to absorb the toxic fatty acids, whereas the cephalexin is an antibiotic which works as a limitation factor. Like it would only allow a certain specific bacteria that we have to check for. So if it passes the cephalexin uh, test, we can use it. So that's the answer. And for Saloni, why do we add charcoal in the transport media? So I did read a bit about this because I had the same doubt, but it had a bit of different answers in different pages. For Thermo Fisher that I had read, for the Jones Kendrick I had read from Thermo Fisher, it had explained that charcoal is used to absorb the toxic fatty acids, and it absorbs peroxides so that peroxides don't come in contact in the medium. And somewhere else, I had also read it that the charcoal is inhibiting the fastidious bacteria by not giving them enough materials.
That's a good question. I think you all should read about it, and when we start discussing medical microbiology experiments, we will be speaking about this. But I think it's something that you guys should read as to why we make use of charcoal. It's, it's a very, very good question, Saloni. So please read that. It is very logical and in the lines of what Akhil is saying, but थोड़ा और brush up करना है, ठीक है? Okay, so I have few questions for everyone over here. Uh, I would start with Akshay. Akshay,
Yes, sir.
you said that you have to divide the samples into different vials so that you can perform all the tests simultaneously, right?
Yes.
So, how would you plan to homogenize the sample before you divide it into the vials?
So basically, like I have two, three like techniques. So basically, we will when we get a sample, first of all, we will have to check for our interest of cells. We will also have various human cells also because after all, we are taking a swab from the swab. So according to me, like not sure, but two of the techniques which I found could be the done. Like first of all is the filtration. Um More than the filtration, I also read about the microfluidic filtration. So what it basically does is we can sort the cells first of all of our need, and whatever the cells we have, then we can mix with the media. Then we can use it. Like for homogenization, we have to basically make all the cells evenly distributed. So after the cell sorting all is done, we can take the cells. And the second thing
Akshay,
might be
I'm not
hello, sir.
Akshay, can you
Yes, sir.
quickly repeat, please? I lost connection.
Yes, sir. So, sir, I was telling like one of the methods like filtration or something. I read about the microfluidics filtration. So basically, what they do is they have a very small wells like structure, like not wells like holes, porous like thing. So they will like we can sort the cells on basis of what the cells we want. So after sorting the cells of our interest on the sample, we can store it, or we can go for the further process. For homogenization, like we have to just we can simply like vortex it. We can just make the distribution of the sample which we have just evenly to be even, and then we can carry out certain things. And the other thing was for homogenization, I thought of cell counter. So I don't know if it can come here, the fluorescence assisted cell one. So basically we can like attack like tag some antibodies basis on the cells we want, and we can sort the cells and we can collect it and we can go for the diagnostics.
Okay. Okay. One question that I have for Sarvesh is that you mentioned about collecting ciliated columnar epithelial cells.
Yes,
Why
sir.
would you want to collect that?
So basically, when we look at Streptococcus and Chlamydophila species, they tend to reside under our cells. So collection of those cells which are susceptible to those organisms might help us to obtain those organisms, even viruses. Yes. Thank you, sir.
Okay, so after you collect or rather while collecting the swab, are there any specific precautions that you suggest?
Precautions after the collection of the samples, they must be stored in appropriate media, and they must be duly labeled with the patient's name
During the
and
collection, sorry, Harish.
sorry,
During the collection.
during the collection of samples. Hello, sir. I couldn't hear you clearly. Can anyone hear Sachin, sir?
No, sir.
I think so. It's time.
Okay, okay, fine. I think Nitin sir, you want to ask questions to anyone, please put it in the chat box if the problem is resolved. Nitin sir. Okay. Harshali, there is a question for you.
Sir, I couldn't get the question. Like, may interfere in. Yes. Sir, I couldn't hear the question.
Question is in the chat box, Harshali.
Yes, sir, but like, which analysis? Like, I couldn't get the question. I'm sorry.
No, if there is going to be mucus in samples, is it going to
Hmm.
interfere in your microbiological analysis? Is the question. So how would you deal with it if you feel that that could interfere?
If there is a presence of mucus in the sample.
Yes. Yes.
Do you think that there will be the presence of mucus in some of the samples? Yes or no?
Yes, sir. No, nasal swab when all taken, they can be.
Okay, so do you think they will interfere or not? Yes or no? And if yes, then can you just give any pre-process?
Caution. Mm, Fine.
Okay, till the time Harshali is thinking, Sarvesh, you can answer the question put for you.
Yes, sir.
Yes, I'm typing it in the chat box.
Preferably answer it out, that would be better, Sarvesh. It will
So,
save time.
flock foam are generally gentle to the inner parts of the patient, and also they are more they have more absorptivity, so they are normally preferred.
Okay. Preksha, are you there?
So
Yes,
maybe we
sir.
can use some chemicals.
Chemicals like what, Harshali?
Uh, Specifically, I don't. There might be like antibodies also, so mucus can come. We can make use of some chemicals to deal with it.
Okay. Preksha, question for you. So, when you say that, there are multiple precautions that have to be taken in terms of collection, transport, and processing, which one of, these steps do you find as the biggest hurdle in terms of upper respiratory tract samples? Which of these
Sir,
is the
like,
biggest hurdle?
sir, we should store that particular sample for at a particular temperature. So that is the biggest challenge.
Any specific temperature that you know of where the
I
sample should be preserved?
I think 2 to 7 degrees Celsius.
Okay, that's it from my end. Akshay, I would request you to also convert this poster after you make the addition of the department and institute name to a JPEG file and
Okay,
email
sir.
that JPEG file to me separately.
Okay, yes,
It will go on the website.
yes.
Harshali, another question for you.
So that was not my part, I guess. GA is.
It's in the diagnostics part. Yes.
So should I answer? Maybe.
Yeah, go ahead.
Yeah. So
Yes, yes, go
G.
ahead.
Yes, sir. So GAS is basically a group A streptococcus. So as we already know that according to the, if you consider the Lansfield group, the streptococcus species, the genus within the genus there are a lot of species which has a very different antigenic properties. So based on the antigenic properties, we can classify it A, B, C, D, E, or many things. So group A streptococcus is GAS. So it is one of the major organisms, which can be very lethal in the upper respiratory tract. So it can cause pharyngitis, strep throat, which is a very common thing. So we can detect the GAS by like for various examples like antigenic strep test, as we know. So we know that GAS has streptococcus pyogenes strain. So we can use the antibodies which are like basically belonging to against that antigen, because we know the antigenic substance group A. So we can use that and we can detect that.
Okay, okay, thank you.
Thank you, sir.
There is a question by Siddesh in the chat box. If any one of you wants to answer it quickly, this will be the last question that we'll be able to take.
So according to me, like definitely the medium cannot be the same. For now, the process has become so high that we get a sample collection media, and in that media only there are a lot of medias which are available. Sample collection kit. So along with the swabs, you also get the instruction for various media. So as Akhil mentioned about various transport media, so if he wants to add anything, he can say. But yeah, definitely we can't use the same one. So we have to collect the sample in such a way that it will like we supposing we have a patient in front of us, so we will know what are the symptoms of the patients. Okay, so we will get an idea. The physician will get an idea first of all, ki what infection can it be? Can it be a viral infection or can it be bacterial infection? So based on that, he will instruct the patient or the hospitals that we have to collect the sample, that we will have to take this sample, so please collect it, so we can collect that way. So if so definitely that's the reason. So if Akhil wants to add anything regarding the transport media for bacteria virus, and he can do so.
No, that was
But
really.
is Siddharth's question regarding processing or transport media?
Sir, yeah, it was about processing. Like Akhil said that we can use that media, universal transport media for viruses also.
Yeah. Then your question is not about processing media. Your question is about transport media.
Hmm.
How is your question about processing media when you yourself are saying you're asking about transport media, Siddharth?
Uh,
And you have probably lost this understanding that a transport is multiple organisms. So how is it really going to matter if it's a bacteria or a virus? Is my question.
Sorry, sir, but I like there was a lot of pain in your voice, so I couldn't hear the last words. Like you probably, and then it was pinging.
I said that if you use transport media is to make viability of the organism, right? So how does it matter if it's virus or a bacteria? We are not providing it any growth medium here. Are you able
Yes.
to hear me now?
Yes,
Yes,
sir.
sir. Now I understood. Because when he mentioned it, he said like we can use that media for growing it. So I got confused. That okay,
Bacteria
how?
is never used to grow. Transport media is only to keep the cells viable, right? And when you talk about growing viruses, you never use a media. You have to use cell lines for that. The media, the viruses are surely not going to grow, right?
Yes, sir.
Yes.
Okay, thank you, group one. I think it's time for us to switch to the next presentation. So you can stop sharing your screen.